Okay, welcome back, everybody. This is lecture 5, I believe. Hard to believe how fast the semester is going. Today, I'm going to try to talk a little bit about cell growth and division, and maybe come back to cell sorting. I know that the homework assignment that was due today was on preliminary work on cell growth and division. Some people had questions about it, and some people wanted to see this lecture before doing the homework assignment. So I don't have a new homework assignment for you for today because I want to see how things go. And I'll try to get the homework up before the weekend so people have the weekend to work on it. But I want to see how it goes. I also have to remind you, as always, that this lecture is live streamed and recorded, so please be aware of that. So today I'd like to start out. I'd asked you to prepare a short discussion of your projects. In particular, again, if people are going to connect and work together on projects, it's important to do that. Starting the homework assignment for next week, I'm going to ask people to, on a weekly basis, give themselves a small project assignment for what you're going to achieve during the week for your project, and then write it up. So each week, I'll ask you to set yourself a small task, be graded as a homework assignment, and then give a small progress report, written one, as another homework assignment. Today, we're going to be talking primarily about cell growth and division. Some of that we've done a little bit of before. We'll come back and explore that in more detail. I'll apologize a little bit in advance because I originally was going to spend most of this lecture on doing the cell sorting exercise, because when I graded that, I noticed that some people were a little confused about a few things. And so I did quite a bit of work on that, but then I thought it would be more useful really to focus on getting us through cell division. And so we'll see how much time we have at the end to do the cell sorting exercise. I hope as things go on, those tools will become simpler to use. And I also want to go over one other homework problem from last week that I think would benefit from a little bit more explanation. So maybe we should start out by just going through the updates on student projects. Who would like to go first on that? Joel? It seems unfair to be always calling on Joel, but for some reason you're always at the top left of the screen, so I have to start. Maybe next week I'll start at the bottom right. So.
It's okay. I can go first. I am first very happy that me and Jackson, we got together and we talked a little bit about the project. James and Jackson knows a little bit of the problems. I think I should pitch to everyone the problems that there is in this model. So if someone has a good idea, we can probably implement something on this model. Let me just share my screen, I suppose. It will be a lot of the same. But wait, just a second. Yep. Oh, can you see this screen? Okay. Yeah, so there is this model that we were trying to replicate from this paper. The problem is we don't have some informations on this model. We have a lot of information on how things work inside the cell, but not exactly, for example, what kind of bacteria is this one? How much PAMPs it is actually generating? And what is the range of interaction here? How much of the PAMPs actually interact with the toll-like receptor for? And other things like the working, is there any kind of communication between these two cells for the more generation of mucus? So it's a little bit of an open question in a lot of points, right? So I found this paper that talks a little bit more of the types of bacteria and viruses that were found in people or in babies with NEC. So one of the ideas that I was having with Jackson was that we would probably could expand this model by associating different bacterias and see which ones are more damaging to the already done model by Dr. Ann and his crew. Uh, Another thing that I was thinking is trying to bring it more to the mechanical part of the system. I read somewhere in another paper that vasoconstrictivity and hemodynamics are correlated to the presence of NEC. I still have to read a little bit more how I would implement that and where. But the idea is also maybe we need more cells than just these two types of cells here, since we know that the guts have more cells than these two, and they might be involved in the generation of the neck. So there's a lot of open points that we can look up. Jackson, if you want to say something about it, feel free.
Yeah, he covered almost everything. The only thing that I found so far with bacteria is that it seems the only bacteria that causes the PAMPs increase is gram-negative bacteria. But that's really all I have to add right now.
Nice. Yeah, thank you for that, by the way. I know that we have like a short time since the last time we talked. Yeah, and so we start generating a lot of other hypotheses. We are actually on the hypothesis generation part of the model that we read. So we're trying to think, wait, is this the first milk that the mom give to their baby, right? I think it has a specific name, colostrum. Does
Yeah.
that has any effect in the activation of the guts from the baby, right? Maybe create something. We know, I read papers saying that the milk can protect against NEC. It has something that helps to protect the toll-like receptor. So there is a bunch of open and loose ends that we have to focus and choose which ones we want to pick.
Well, I think I mean, the gut bacteria system is extremely complicated. And you bring up in neonates, there's this additional complication that their immune systems work differently from adults. I mean, the antibodies that the baby has typically come from the mother, because it takes time for them to be generated by the baby. And so there are a whole series of things. I hope you'll be able to join. I think we're scheduled for a phone call with Gary later in the week. And I don't know how much time there'll be in that call to discuss this project, but at least you'll get to meet him and maybe if nothing else, schedule a meeting to go over that paper. I think the issues you're raising are fascinating. We'll see how far you get in a semester. I think replicating that paper by itself is already going to be pretty challenging. And so we'll have to pin him down and let's see how he responds. But we do have the appointment, so that's good.
Yeah, and if we're thinking on like using this model to get modules out of it for the immune system and things like that, we could focus more into that, choosing future models too. That's another thing.
Well, since Claire Bryant at Cambridge is interested in salmonella, particularly in gut bacteria, then potentially this is a good choice for a model to start with. So the key will be figuring out what he did, and then implementing. That's great. Well, I'm delighted that you're making progress. It's a fascinating problem. Okay, Alex.
Yes. So I figured out what I'm going to be working on. So after talking with TJ, we came up with he gave me a list of like feature improvements essentially that he has ideas for in CompuCell, and the one I'm going to focus on is working on this field extractor class. So like the big problem with this is that this runs on every time CompuCell redraws this like graphics field. So it runs a lot, but it is not implemented optimally. Well, like it's a serial implementation, which is probably decent enough, but the fact that it is a serial implementation is the problem, because just on this simple little thing, it's a 200 by 200 field, so it's looking at each point, and so every attempt to redraw this is an n squared, and then if it's a 3D model, it's an n cubed operation, all of which is serial. So improving this to make it parallel using OpenMP is the first step, but that's not going to be easy because it's written serially, like and implicitly. It's like it does these insertion things, which is not thread safe because it assumes order. So I'm gonna have to rewrite a lot of this code to work around that. And then depending on how long that takes, the next stage after that would be to try and work on a CUDA accelerated version as well.
That would be lovely if we could. These days GPUs are powerful, and not taking advantage of that, especially for the graphics, is a pity. I think
I haven't looked into that as much, but he mentioned that because you use a library called VTK to do the rendering for these things, and he mentioned that you can use the like talk to it directly from the GPU and skip all of that data transitioning as well.
I would have thought VTK had some GPU enabled modules.
I just haven't looked because that's like later on.
I'm not. I mean, it's fine. I'm not being critical. I just.
Yeah, I'm sure it says something.
They're a reasonably sophisticated company. We might even be able to talk to them if there were issues about implementation. They might be willing to talk to us.
Yeah,
I know TJ did have a meeting with them a couple of months ago, and so that's possible. But yes, the imaging and display in CompuCell is slow, and there are even things that are simpler than that are crazy, which I've never understood. Which is one thing that's slow in CompuCell is actually print statements. I don't know how a print statement can take time, but you put print statements in your Python, and it dramatically slows down the execution of the simulation.
yeah, that's because CompuCell is, well, I can assume the answer for that if you want me to get into it, but
Sure.
It's not really needed. I mean, it's the classic issue with any, because CompuCell is really just running Python. So like this command prompt is CompuCell. So every time you print whatever, and most users ignore this, it has to take time to print those characters to this screen, which is relatively slow compared to like computation.
I was shocked how much of the execution time is actually dumping to the console.
Yeah,
that was not a problem in Fortran. And so I guess the old days had some advantages. Well, that's great. I think that would be really nice. I know that refactoring the code to be able to be parallel will not be trivial. Although, at least here, there shouldn't be dependencies in the sense that if you're using stale parameters to do the display, it doesn't change the state of the simulation. So you're may have issues with state of the image, but it shouldn't affect the simulation.
Yeah, it's gonna take some time to understand all the code paths, because it goes in and out and calls several functions to get all the information.
Yeah, it's interesting because the core algorithm for the updates, the lattice updates, is actually in OpenMP, and
Yes.
and so that is parallelized, and that algorithm is quite powerful. It works very well, although it has some pathologies. It's possible to crash it, and we may encounter that. Typically, execution speed isn't that critical for us here, so we don't turn it on much. But actually, even in NanoHub, if you ask for 4 processors, it'll give them to you, and it will run faster. Of course, on your desktop, if you have a lot of processors, then it can speed things up a lot.
That's all I have.
Great. No, that's wonderful. Okay, I guess you'd see Connor.
So Andrew and I threw a few slides together, so I can screen share, and Andrew, when you take over, you can just tell me when to click forward. Can everyone see if I put this on full screen? Okay, wonderful. So for this semester, we are looking to, at a baseline, replicate a paper that uses the cellular POTS model but doesn't implement it in CompuCell 3D, except we're going to make a couple of variations to it. So generally, very broadly and abstractly, there's a lot of unanswered questions when it comes from that transition point from unicellular life to multicellular life. And one of those is that even though cell-to-cell adhesion is important, the more integrated you have groups of cells become, the more trade-offs there are. And a lot of research to date, theoretically and simulation-wise, that works with these things will kind of implicitly assume an advantage for multicellularity in the fitness function, or they'll try to encourage that behavior to study dynamics about it. What that doesn't answer is in what conditions we might expect to see some of these transitions take place. And that's what this paper that we're going to talk about kind of tries to begin to answer. So on the right here, this is the main conceptual graph from the paper, and they have a couple of primary elements. One is that they have adhesion, where there's ligand binding sites, and basically all the cells can bind to each other, but the strength of that adhesion is determined by the way that the bits match. So here we have 1 and 0 causes an increase in strength, but 0 and 0 and 1 and 1 do not. There's also persistent migration. So as the cell is moving, it tends to continue along certain trajectories it's been on. And the really cool thing that they had about this is that they have a non-diffusing gradient in the environment where it's patchy. So you'll see little white specks if you look closely, and that's points where the gradient has been punched out. And basically what that results in is that if you're a single cell chemotaxing, it's really hard to climb up the gradient because both you have the persistent migration and there's noise in your signal. And what they ended up finding in this was that they implemented seasonality. So basically, every certain number of steps, the source of this gradient would move. And for long seasons, they saw a push towards multicellular strategies where these binding sites evolved to become stronger. And in very short seasons, they saw unicellular strategies. And at intermediate seasons, they saw an evolutionary biostability where you could get both. And the reason for this is that if you have a long season, you can chemotax more efficiently towards the gradient. But in short seasons, you don't really want to go up that gradient super explicitly because you're better off kind of ending up close enough up it that you can reproduce, but far enough away that you might be where the next fitness peak appears. So one of our goals is to implement the variation of their model, and Andrew's going to talk about where these variations are in more detail, but that tries to capture the effect of the different periods of seasonality, the speed difference between the multicellular and unicellular aggregates. And if we finish the replication on time, we are going to try, we're still figuring out, but we're considering a couple of additions we can make to the model. And Professor Glazier, I don't know if you saw, but I think yesterday we got an email back for a meeting with Merckx about discussing it more. So that is all good to go. And all right, Andrew, you can take over now.
Yep. So I'm going to be talking about the inputs and the controls, and then the conceptual model. So describing what is the biological basis for what's being implemented in. But the next slide. So the general idea is that multicellularity seems to be more advantageous than having individual cells moving, doing their own thing. And the interesting thing is that this is not the goal of cells. It just so happened that because they were able to come together, they had higher fitness and were selected for those conditions. And so this is something that we are keeping in mind when we are evaluating how well our model is doing. It's not about how many of the cells are coming together, but rather how much of this chemical gradient are they consuming individually. And then the idea is that through this evolutionary algorithm, we will be able to, through this evolutionary algorithm, the cells that are able to consume more of the gradient are able emerge to actually moving collectively. Can move to the next slide. And yeah, so now the conceptual model. So they're implementing the cellular POTS model, and we're gonna do that with CompuCell. There's the cellular migration where based off of Beltman et al., a cell can walk consistently in a single direction, but then it will stochastically move to another one. And with cell replication, the cells divide on its shortest axis. And cell death, so after the replication, half of the cells are dropped off at random, and a new season is started where the gradient is in a different location. Go to the next slide. And so with how they did chemotaxis in their paper was that they averaged the level of chemoattractant concentration covered by each cell's area. But we're going to do it differently whereas the CPM model, a lot of the behaviors and attributes that are implemented are based off of minimizing energy. So what we're going to be, how chemotaxis is implemented in CompuCell is it measures the difference, how much lower energy or how much minimizing the energy from where it wants to go and where it is. And if it ends up lowering its current energy state, then it will do that action. I'm going to go to the next slide. And the evolutionary dynamics are also slightly different, but generally we will evaluate their fitness by measuring how far away the cells are from the peak, and then what is mutated during this evolutionary run is how much the ligand binding for cells adhering to each other changes. So as Connor mentioned, they had bit strings, and the bit strings that were the most complementary bound were able to bind the best. And for our replication, we're going to have each cell have an integer from 1 to 6, and then the lowest number will indicate its binding strength. And basically after a certain number of Monte Carlo steps, the season is over and we evaluate the fitness and we will assign a chance of reproduction based off of that, and then mutation takes place. And I guess we're going to walk through a timeline of how we're going to go through it.
Yeah, I'll do this quickly, but generally the goal is just to have an optimistic schedule for replication in hopes that we will be able to get it done with the extra time if we need more wiggle room, and we could do something in addition if we have that extra window. So to begin, taking an existing chemotaxis simulation and measuring the net speed as it goes through chemotaxis, testing, putting noise in the gradient, and then going ahead and assessing how changing the size of the single cell along with other parameters has on the speed, because that should kind of map on to the multicellular aggregate where the cells are smaller. Then for the next week, we are going to implement seasons within this single cell simulation to get the gradient to move around at different periods. Week after that, and these are some figures on the right from their paper, just to give an idea of what we'd want to expect. Implement the ligand binding site, so this is our transition to multiple cells, reproduction and mutation, and then having graphs for the speed for the individual cells into collections in the case that they make aggregates. And for the last week, implement persistent migration, and this would be where that would leave us where we can start playing around with anything that's different about the model and adding in some extra details.
Yeah, the general idea is that if this takes longer than what we have mapped out, at least we have something solid, and we can finish up with that. If we're finishing on time, we have a lot of room for going into really interesting areas.
I think it's a great platform for exploring these kinds of evolutionary questions. And I saw Roland's email. He said he didn't think that the specific type of chemotaxis was important, which is a relief. If we don't have to implement that, that makes life a lot easier. As I said, I think doing the recognition of the surface receptors is certainly possible. It might take a few hours to implement, but it's not terrible. And the directed motion is also not hard. Giuliano is very expert at giving you a persistence time that you want using explicit forces. So we'll probably cover that. I don't know if we'll do it next week, but we'll cover that in the next couple of weeks. We'll definitely get through that. And we'll certainly start talking about chemotaxis and chemical fields quite soon. So that should help. So in a sense, you're ahead of the curve on that. So maybe you can give the lecture on chemotaxis. But I think it's interesting, and certainly if Roland is engaged, that's a great opportunity. I think certainly for Joel and Jackson and for the two of you, if you could get some coaching from Gary in one case and Roland in the other, that's definitely a bonus. Alex working with TJ a bit. So these, as I've said many times, these are complicated problems, and so getting more help and more inputs is great. And so we definitely should be sitting down periodically and going over this one-on-one, but I think it's very helpful to see it as a group as well. Okay, so I guess Josh.
Just one second. Okay, so my proposed project is a slime mold simulation, specifically, just skipping forward a bit, I already have the code. It was already implemented in CC3D, although the paper was written back in, I think, 2014, so it's most likely a very old version, so I'd probably have to redo the whole thing. But I guess since it's already implemented in CC3D, I would try and make it interactive to be placed on nano hubs. So like you could draw barriers, place food sources and things to stay away from, and things of that nature. The source material, the main one with the code, is this Emergent Networks of Slime Mold Simulation. It was a bachelor's thesis. And then also I kind of wanted to see also for the purpose of just seeing how well my model works, is comparing it to the results of this paper, Rules for Biologically Inspired Adaptive Network Design. This is the very famous one where they grew a slime mold to replicate the Tokyo transit system. So biological context and significance, I'm not really a biology person, so I have to really read up more on the biology of slime molds. I don't really know how they work, but at the moment, after watching a couple of YouTube videos, they are capable of moderate levels of intelligence and are very basic organisms, so it's pretty interesting. And just, I guess, a preliminary hypothesis, draw a maze with 4 paths to a food source and see if it can find the shortest path. And that's all I have for now.
Okay, that's fine. I think I'm glad you were able to get the source code, because it was a little worrying to me given the non-description of the model in the paper. Without that, it could be challenging.
Yeah, I guess that's another thing that I could add, is that bachelor's thesis, like you said, we went through it together and it wasn't very specific on the actual model. So hopefully my write-up will be able to do that a lot better.
I mean, that's something that actually came up in the fall, although we wound up not acting on it, which is that every year we have students who generate these projects, and some of these projects are very finished. Some of them people run out of time. And some of them are great ideas. And so I think next year, in the fall class at least, I'm going to encourage people to think about taking a project from the previous year and continuing it. And this is something especially maybe for our auditors. If you're not going to do a project this semester, think about, as you watch people doing these projects, think about how these ideas could apply in your problems. And perhaps our auditors, who, especially experimentalists, could think about proposing problems that could be solved. And even if they don't get solved by this class this semester, we're going to have our CompuCell workshop, which will cover all of the material in this class in a week in July. And so perhaps, and those summer classes have a hackathon during which people work on projects. And so it might be interesting to come to that and propose a couple of problems, and perhaps recruit some people to work intensively on those problems. So even if you're not going to solve the key problems you need this semester, you're welcome to do that, and I'm very happy to help you if you decide to do it. But perhaps we can think about this as a dry run for that, seeing how this works. So if the three of you have anything you want to add to this, to comment on, you're welcome. You're not obliged to, but if you have anything you want to say now, you have the same privileges as anyone else.
I actually just got out of a meeting in which we were discussing the best way to frame my existing project as like a computationally valid question. So I will get back to you on that. I think I have a couple of things that would be really interesting for people to maybe take a bite out of, if not this semester, then maybe the next.
Well, so why don't we get together? Why don't we try to set up a time to meet one-on-one, or a few on one, and go over that with you. And then if you want to present that material in class also, that would be great.
Yeah, sure.
Okay. Because I know some of the problems I've been hearing from Embryo are very good problems for CompuCell. Some are maybe more difficult, but it would be really interesting to think about those together. All right. Thank you, everybody, for doing the presentations. I appreciate it. We'll keep working on that together. So I want to just review a few of the key ideas that we had from class last time. One of the key things that we did last week was begin to use Python to specify cells' target volumes and target surface. In the CC3D-ML, you take out all of the detail in the volume and surface plugins. And that means that in the Python, you have to supply that information. So for example, here, we've gone over all the cells of the simulation and given them a target volume and a target surface, lambda volume and lambda surface. And we'll be doing that again and again as we go through this semester. Another key concept that we dealt with, which we will have to, which I hope for people who are not heavily computational will become familiar relatively quickly, is the idea of dictionaries. Sorry, I'm having trouble with my windows here, so let me just give me 1 second to reorganize my windows. I tried to advance the slide, and instead I managed to clobber the screen layout. The key things that we're going to use all the time are the cell dictionary, which is a place to store states of cells and information, whether biologically meaningful or computationally useful. And a key thing about Python dictionaries is that the dictionary elements can be heterogeneous and they can be complex objects themselves. So a cell dictionary can have a dictionary inside of it, or a list inside of it, or a number or a string, or some combination of those. And we're going to use that dictionary all the time. Another thing that we'll run into starting today is that when you have multiple steppables, you have to get information between steppables. One way to do that is to use globals, which are not widely encouraged in programming these days. And so there is a shared dictionary, which probably should be called something like simulation.dict rather than shared _steppable_VARS. It's an awkward name. But this is a place that you can store information that is then accessible from everywhere in the simulation, whatever the steppable. There is some computational cost to the use of dictionaries because of the way they're keyed. And so, as with all Python, there's some time costs to using it, but it's going to be quite convenient. One thing you do want to watch out for is don't say cell.dict, quote, cell name equals, quote, equals cell. The one thing that you can't store in these dictionaries are these handles that point to object attributes, objects. So you want to be a little bit careful about that. I know that the hard way because I remember somebody doing that and spending weeks trying to track down the bug because it looks like it should work. Okay, we also talked about tracking fields, which is one of the first things we used a dictionary to do. If cells have an attribute in a dictionary, a single line in the init allows you to color the cells in their display with the value of that attribute. And we're going to use that repeatedly today, and it was in the homework as well. It's possible to have vector valued attributes, and I've never tried it, so somebody should try it and show what it looks like. We also talked last time about plotting histograms. Histograms can be quite useful. We're more likely to be plotting time series because those are the things that we often respond to visually, but histograms also, if you're doing evolution, you care about distributions of characteristics of your organisms, and so histograms will be important. Histograms function very much the way regular plots do. In fact, the creation of the histogram uses the same initial function to create a plot window. Add new plot window is identical, whether you're doing a time series or a histogram. It's what you put in the plot window that's different. I'm trying to get somebody to write something which will pop up a plot window that is a text box, so we can display text while we run. In particular, on NanoHub, the console output is hidden, and so print statements in the Python don't show up when you're using NanoHub, which is an annoyance. I also talked a little bit about some of the things that you can do. Not all of them work perfectly, but you do have in the, if you hover over a plot window, whether regular plot or histogram, right-click on it. There are a variety of options. One of them is export, and it allows you to both save images and save data sets, and that can be quite useful. It's not that you can't open files and save data as files, and that's really a better way to do it, because then every time you run the simulation, you get the same thing. But it's also cumbersome to remember path names, figure out where the files are, and how to read them. And so having a quick and dirty way of saving data and saving images without having to write code is actually very nice. So there's this export option. You can export images or comma-separated values. Either way, you can save it. And if you do that on NanoHub, there are issues with downloading. Here, it's easy if you're on a desktop machine. So those are things to remember from last time. Something that I talked about a little bit last time, and actually I think in the first or the second lecture, and that we might come back to depending on how far we get in class. It's more of a convenience thing than anything else, is that when you create the list of cell types in the CC3D-ML, you'll notice that all cells are given cell type ID and a type name. And that type name will be displayed in various places, and the ID will be displayed in various places. If you type cell.type, the attribute type returns the ID, that is the integer number associated with the cell type. If you type self dot and the name of the cell type in all caps, by all caps, so medium in capital letters, dark in capital letters, that will return the type ID, 0, 1, or 2. What's missing is the ability to get the cell type name as an attribute. And you can get it for an individual cell. There's a function self.getTypeNameByCell, and you can call that for a cell, and it'll tell you the type name for the cell. That's fine. However, suppose that you're writing a simulation in which the user may change the number and names of the cell types. And so you don't know that the cell types are going to be dark and light. You don't know if you're going to have 3 cell types or 5 cell types. You don't know what those names are. In those situations, you might actually want to be able to inquire how many cell types you've got and what they're named. And there is a function to do that. And when I was reworking the cell sorting code, I was trying to do this so it automatically labeled the graphs with the names properly. It's not hard to do it, but it does take a little bit of Python fiddling. There is a utility called extract type names and IDs, not very elegantly named, which will return to you a list which is the list of type names as a function of the number. And so if I say type name dictionary equals CompuCell, da, 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 da. I can now iterate 0, 1, 2, 3, call type ID, and that will return the name. Why would I want to do that? Well, if I don't know what I have in my system, this allows me to introspect the simulation and inquire what it's got in it. If I want to know how many names there are, len of type name dict will tell me how many different types I have. It'll actually give me the number +1, which again is rather Pythonic, because everything's zero-based and loops end one before the end. So that's something that we might remember. If you don't remember it and you need it, ask somebody, because it's not something we do every day, but it is something that, specifically in that cell sorting simulation, it's something that's convenient. As I mentioned, if you're running on NanoHub, print statements are not going to be visible, and we're going to try to update CompuCell so that the console outputs are actually displayed. I'm not quite sure why that version doesn't do that, but it does. I wanted to come back now, having graded the homeworks, to this issue of neighbor order and contact energy, because this is a little bit confusing. And I gave you a homework problem, the second homework problem for last week, that was reasonably tedious to do by hand. And the tediousness of the exercise was partly to illustrate some of the perhaps counterintuitive things that can happen in lattice-based computations. And also, I think once it's important to do those things by hand to get a sense of how they work. And so if I look at contact energy between two cells, I'm summing over all cells and all the neighbors of the cells. And then I'm asking the question, is the value at will site the same as the value at a neighboring site? And the issue is that the neighborhood is not necessarily the nearest neighbor. So if I look at this boundary between gray and white, and I'm doing nearest neighbor computations, then the boundary length is just the number of edges between gray and white. So I have one, people can see my cursor? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 edges between gray and white. So nearest neighbor, it's straightforward. If I go to longer ranges, it becomes less straightforward. And next nearest neighbors are the square. That's still not so confusing, perhaps, with the diagonals added. Third neighbors include the same direction as the first neighbor, but one further out. Fourth neighbors, and so on, going up. CompuCell has the ability to go to arbitrary neighborhood. If you tell it to go to 27th neighbor, it will. Slow things down a lot. The number of neighbors of each order is in this list. The scaling is not obvious. And it's different in 2D and 3D. When I do contact energy, I have to find all of the mismatched connections. So if I start at site I, I'm in this gray cell, and its nearest neighbors, I have 2 mismatched nearest neighbors in the white cell. If I go to second neighbor, that adds 1 extra voxel. If I go to third neighbor, it adds 2 more. If I go to fourth neighbor, it adds 1, 2, 3 more. If I go to fifth neighbor, it adds 1, 2, 2 more. Sixth, 2, 7, 1, 2, 3, 4. And so how many I add will depend on the shape of the cells in a sometimes non-obvious way. Each one of those gets multiplied by whatever the J is between gray and white. So the homework problem 3.2 gave you a picture. Here I defined 7 cells plus the medium, and the cells were of 2 types, blue and red. And I gave you a set of energies. J medium blue is 5, J blue blue is 15, J medium red is 5, red blue is 10, red red is 8. And the first thing I asked you to do was to copy down that adjacency picture, which people needed. And I think everybody was able to do that. And now I would like you to, let's do this together. We are just going to pick one cell. We're not going to do this for all the cells in the pattern. If you did it, you'll remember that it's tedious to do it. But let's do for neighbor order 1, and let's just pick cell number 1 and ask the question, what is the contact energy for cell number 1, assuming nearest neighbor interactions, neighbor order 1. So why don't people take a minute to do that, think about it, and then I will walk through that calculation. And I think people generally were able to do this for the nearest neighbor 1, but doing it for the longer ranges was somewhat confusing. I want to go walk through that with you. So again, the way I would do this is that you are going to count how many mismatched links there are. And again, you have to remember that you only count bonds between different cells. And some people got a little confused about that in the homework. And I'd count separately how many red-blue contacts do we have, how many blue-blue, and how many medium-blue. There are no red-red because my cell is blue. And so if I look at my cell here, it has 1, 2, 3, 4, 5, 6, 7, 8 units of surface. And if I go around, I have 1, 2, 3 units of red-blue surface, 1, 2 units of blue-blue surface, 1, 2, 3 units of blue medium surface. And now to get the energy, I have to multiply those by the appropriate J. J red blue, 10, J blue blue, 15, J medium blue, 5. So I'm weighting the energy per link or per bond. 3, 2, 3. 3 times 10, 2 times 15, 3 times 5, 30 plus 30 plus 15, 75. So the energy, surface contact energy associated with cell number 1 is 75, and I have to repeat that for all 7 of these. Okay. The next one is a little bit less friendly, which is let's do the same thing with third order interactions instead of first order interactions. So now I have to very systematically go through, put my stencil here down on top of 1, each of my cell voxels. And once I have the stencil, I have to walk out to the third order, check, and each time ask the question, 1, am I inside of the cell I'm already started with? And 2, if I'm not, what's the type of the cell I'm in? And count. And so let me just do that. I'm not going to expect you to do it by hand as we do it. It took me a while to do the graphics, and I hope I didn't make any mistakes. Okay. And I'm just going to do it for one of the voxels. First, we did it here for nearest neighbor, we did one cell out of 7. Okay, I'm going to do one voxel out of 3. So if you're going to do it for the whole thing, it's rather tedious, but we do it systematically. So I start at this voxel here, the top right voxel number 1, and I find the nearest neighbors. And the nearest neighbors are circled here. And I'll notice that one of them is inside my own cell, so it doesn't count. One of them is inside a different blue cell, so I get 1 unit of blue. And 2 of them are inside cell 6, which is red, so I get 2 units of red. So if I walk around the edge, 1 unit of red from this 6, 1 unit of blue from this 2. This one doesn't count because it's in my cell. And this 6 gives me another unit of red-blue. So after I've done nearest neighbors, I have in my counting 2, 1, 0. Now I go to second neighbor, corners, these 4. I do the same thing. I start here in the top right. I have 1 unit of blue from this 2. I have 1 unit of medium now from this corner. I have this doesn't do anything because I'm inside my own cell. And then the last one gives me a unit of red-blue. And so I've got 3 more voxels, 1 red-blue, 1 blue-blue, and 1 medium blue. I now go to my third neighbor. I've now jumped. Notice that it's possible here. I'm going from cell number 1 to medium, crossing over. I'm hopping over cell number 2 when I do this. So it's possible to jump over another cell. And so I do exactly the same thing again. I'll start at the top. No, sorry, here. I start at the top. 1 to 2 is blue, blue. Even though there's an intermediate red, that link is blue to blue. So it gives me a blue, blue, 1 unit of blue, blue. The second one, again, I'm hopping over a blue to medium. And so 1 is next to medium here, even though it's not next to it in nearest neighbor sense. And so this gives me 1 unit of blue medium. Going down, I'm hopping over myself. So I get 1 unit of blue medium from this voxel. Notice that that same unit of medium would be counted also coming from the voxel below it and from the voxel below and to the left. All of those would have that same medium voxel as a neighbor. So that medium voxel would actually be counted 3 times in contributing to the contact. And then the last one here is the 6, 1 to red here. Notice again, this 1 is next to red in this first 6, and it's next to red in the second 6. It's also next to red here on the upper diagonal and north. And so this number 6 actually contributes 4 units of red, blue, just for this one voxel, for this top voxel. Okay. And so now I have 4, 3, 3 as my contacts. 4 times 10, 3 times 15, 3 times 5, 45 plus 45 plus 15, 100. And does somebody remember what it was for the whole cell with nearest neighbor? I should probably. Sorry?
75.
75. So this one voxel is contributing more at third neighbor than that whole cell did at nearest neighbor. Now I asked you what you thought about how this scaled, and people said, well, approximately with the distance. And it's not a terrible guess that the typical energy scales with the distance. But it's not anything simple. It depends on the cell shape. It depends on how big the cells are. And And so this is one of the complications of working on the lattice. Are there any questions about that? We're not going to generally do this by hand, but it's important to have a sense of how it works, because when you change those neighbor orders, you change things in ways that are rather subtle. In particular, this ability for cell number 1 to be next to, well, in this case, it's not doing anything. Up to third neighbor, it's not doing anything strange, because cell number 1 would be next to 6, 2, and medium, and 4, nearest neighbor as well. Let's see. If I go to, I don't think this particular layout doesn't do anything strange. But if I look at some of the other cells, going to third neighbor might actually mean that I'm next to a cell that I didn't think I was next to, because I'm hopping over one of my neighbors. And so you can be adjacent to more different neighbors. When you iterate over neighbors to count, you can have more neighbors than you expect. And you can imagine that biologically, if you think of some kind of thin process that's squeezing in between cells so that cells can contact each other even if they're not right next to each other.
I have a question. Is that
Yes.
considered at every time we are trying to do the copy of each voxel? Is when it's considered?
So there are two things going on. That's one of the other things that can be a little bit confusing, is that you have, in principle, the ability to have different neighbor orders for different processes. And so in the basic declaration of the cell lattice, you'll be declaring a neighbor order. And that's for doing voxel copies. So that says, if I try to replace a voxel with my current voxel, how far out can I reach? And the idea here essentially is how far can the membrane of the cell fluctuate in a time step. This is more a question of how big is the interaction range of cell adhesion molecules. And so that's done separately. The neighbor order is specified separately for surface energies.
Got it. Thank you.
So you can use them. It probably makes sense to keep the numbers the same, but they don't have to be.
It was definitely a little bit confusing to me because I didn't know if that was considering during the time of the copies.
So because the copy determines the dynamics, it determines that metropolis algorithm, but the energy is independent of the copy dynamics. And so one thing that you'll see is that as you increase the neighbor order, the contact energy becomes larger. And that means the contact energy relative to the fluctuation amplitude becomes larger, which means fluctuations become smaller. And so in this example, and this was originally a homework, I think not a homework problem, it was originally going to be an in-class exercise, which we're going to skip because of time. If we do nearest neighbor interactions, here we have a cell that's rather fuzzy in its boundaries, although you'll see that there's local alignment of the cell boundary with the lattice. One of the problems with nearest neighbor interactions is that they're very square, and you'll see that as in non-physical alignment of the cell with the lattice. Then there's an intermediate length, and 4 usually is the best for square lattices, where the cell is quite round. And then if I make the interaction range very large, my fluctuations are getting smaller again, and I'm seeing the cell line up again on the lattice, this time because of the fluctuations being too small. This is a little bit similar to changing the contact energy per unit area. N equals 7 is about the same as a contact energy of 50, and N of 1 is comparable to a contact energy of 1. So going from N equals 1 to 7 effectively increases the contact energy by 50-fold. So it's not crazy to think of it that this scaling is n squared, but it's not quite n squared. But certainly, and people notice this, if you sum over a larger neighborhood, you have more connections, and therefore the values are large. On the other hand, the longer interactions also reduce the lattice anisotropy. As I say, nearest neighbors are really square. Second neighbors are a little bit better. Third neighbors don't actually gain you much. Fourth neighbors do quite a bit better because we're trying to draw a circle. And we're doing that with stair-stepping, dithered. And the bigger the radius, the less the stair-stepping affects us. And so here, if we go to longer ranges and adjust the J's so that we keep the fluctuations the same as well, we'll see that as we go to longer interaction ranges, things get rounder. And again, the issue for us is that when you make these changes of neighborhood or neighbor order, it's a little tricky to know how much you have to adjust the energies. That's something you have to explore because it will depend on things like how big the cell is. There's no simple way of getting that. You can get a rough back of the envelope from looking at how many neighbors there are out to radius x, and that'll give you a first order approximation. Okay, so I'd like to spend the rest of the class talking about cell growth and division. This was primarily covered in the homework assignment for this week. As I mentioned, people had, last week when I gave the assignment, I said that people could try it. We'll do some of the homework problems together now. I hope that most of you were able to get through some of it, but there were a few people who had questions and wanted to hold it for next week, and I'm happy to do that. So one of the key things that cells have to do biologically in order to build multicellular organisms, and even some unicellular colonial organisms, is they have to decide whether they're going to proliferate, that is, grow and divide, sit there and survive, it's often called quiescence or arrest, change their identity in some fundamental way, change their type, which is differentiation, or die. And those decisions depend on a whole range of both internal and external signals. And the cells are continually integrating those signals and deciding which of those things to do. And there are other things they can do, like move or pull on things. But ultimately, the fundamental decision a cell has to make is, do I grow, do I divide, do I sit here, or do I die? And then we'll find that there are different ways cells can die, and that those have different effects, although those are not explicitly represented in the models. In CompuCell, those have to be implemented. There are a couple of things that we can make as very generic biological observation, and the biologists in the room will wince at the degree of oversimplification here. And please feel free, all of the biologists, to correct me when I make a misstatement about biology. I'm always glad to have somebody put things right in the biological record. But there are a couple of things that we will run into repeatedly in this class as we go forward. One of them is the idea of what could be called a critical limiting nutrient or critical limiting factor, which is that for a cell to grow, it has to be able to take in both energy and material to build more cell. And normally that energy comes in the form of glucose, although there are other particular cell types that use other energy sources as well and other material sources. The second thing that the cell needs in order to be able to survive and grow are typically growth factors. So cells don't grow and divide in a vacuum. They signal to each other, and growth factors which typically are going to be abbreviations ending in the letters G and F for growth factor, are an enormous family of different signaling molecules, many of which have similar behaviors, many of which don't. And we can look at this picture. I'm sorry about the overlap here, but we have IGF here, TGF, FEGF. We don't have FGF, which is the one that you run into the most commonly in development. All of these are growth factors. These growth factors sometimes will come directly into the cell. At other times, they will bind to a transmembrane molecule, transmembrane receptor, that will then send a signal that promotes cell survival or cell proliferation inside the cell. And so it's pretty common, just the way computer programs typically have an API that's their interface with the world. Cells use these receptors as their interface to the world. And so you can think of these as essentially input channels on a box, which is the module representing the cell. Things are never that simple, unfortunately. So conceptually, it's not a bad map. Cells will also respond to additional kinds of signals. One of them, as I mentioned, I think last week, is whether they're attached to their neighbors. Here we have integrins, which attach the cell to the extracellular matrix. We'll also, this particular picture does not have cadherins or CAMs, which are cell adhesion molecules that attach cells to each other, which also send signals. You'll notice down at the bottom, there are what are called death factors. These are signals that promote cell death. You could say, why do you want to have cells die? And the answer is that programmed cell death is critical to development and maintenance of tissues. If cells don't die when they're supposed to, you have a problem. Cells also are continually monitoring themselves to find out if they're healthy or not, if they're in the right place or not. And if, in general, cells have limited, not zero, but limited ability to fix themselves when things go wrong. And so very often the best strategy for a cell that's damaged is to die, get out of the way, and then let a healthy cell replicate and take its place. In the case of things like infection, in general, once a cell is infected with a virus, the only way to get rid of the virus is to kill the cell. You can't eliminate the virus from the cell. Again, there's some in biology, there's almost always an exception to everything, but in general, once cells are infected, the only way to get rid of the infection is to kill the cell, which is one of the reasons that when you get a viral infection, the thing that's making you sick is your immune response. Feel sick, not be sick, but feel sick is your immune response to the virus, not the virus. And we'll see a bunch of signals here. We're not going to track all of these pathways. We're not going to be doing much with that in this class, although people certainly build models that include this using CompuCell. You'll see some players that will show up quite frequently. PI3 kinase here, RAS, which was originally called an oncogene, which is not. And these are molecules that promote cell proliferation. And you'll also see down at the bottom, there's something called here abnormality sensor. You'll see a molecule called caspase 8, and these point to apoptosis. Apoptosis is programmed cell death. Cells can die in a whole variety of ways. You can smash them to bits in a disorderly way, which is classic necrosis or lysis, or they can actually shut down in a very orderly way, and apoptosis is one of the orderly ways that cells shut down. There'll be a couple of players that you'll see repeatedly if you look in the literature. Caspase 8 is one. P53, which is the choke point for the relaying of cell death of apoptotic signaling. There are a couple of other ways you can induce apoptosis, but if you look in cancer, in solid tumor cancers, and ask the question, what genes are disrupted in most cancers? And the answer is that there are only 3 or 4 genes that are disrupted in more than 10% of cancers. P53 is one, because what really matters is not the genes, but what the genes do. And for most functions in a cell, there are many pathways that do the same thing, or almost the same thing. And so disrupting one of them doesn't necessarily disrupt the others. For programmed cell death, P53 really is a choke point. And so messing with P53 does greatly inhibit cell death, programmed cell death. And that means in particular that damaged cells don't die, which is one of the ways you get cancers. It winds up there actually is a mirror of P53, which can get turned on in some cases as a backup. And famously, Carl O'Malley, who's now at Arizona State, discovered that elephants are extremely cancer resistant because they have many, many, many independent copies of P53. So to knock out apoptosis in elephants, you have to knock out all 20 copies. I think it's not quite 20, it's like 12 copies. Whereas in humans, you have to only knock out one. So we're not going to go into this whole complexity here. Another molecule, though, that I want to mention is beta-catenin, which you'll see here. Beta-catenin is an interesting molecule because, as I mentioned the other day, in engineering, we try to build things that are orthogonal, so that when you change one thing, you change that thing and not other things. Biology, because it's not designed, does not have that kind of engineering principle at work. And many molecular species and structures play many roles simultaneously. Beta-catenin is particularly interesting. It's not shown in detail in this picture because it is a structural molecule. It connects the cell cytoskeleton to the cell adhesion molecules on the surface. And so it helps cells stick to each other. It also plays a regulatory function. When it comes off of the cell membrane, it sends a signal promoting cell proliferation. And this allows cell adhesion molecules to essentially sense whether the cell is in the right environment or not, and whether there are enough cells around the cell to make the tissue, to keep the tissue intact. If there aren't, if the cell's in the wrong environment, it's going to tend to make a decision to die. If the cell is in the right environment, but there aren't neighbors all the way around it, that beta-catenin signal will promote cell proliferation until there are. And so this is a particularly nice way of regulating the density of tissues. Integrins play a similar role. They detect if the cell is in the right extracellular, not cellular, but extracellular environment. If the integrins are bound to their targets, the cell is in its right place. If they're not, the cell has somehow wandered off into the wrong place. And that actually will send a signal through caspase 8, which is not cooked up here in this diagram, through caspase 8 telling the cell, wow, you wandered into the wrong place, maybe you're metastatic, die. And so there's a very strong cell death signal from integrins if they're not bound to their targets. So all of this complexity is beautiful. Today, we're going to ignore all of that complexity and talk about very, very simple things. But I wanted to talk about this at least a little bit because if you want to think about the principles of organization, particularly how you build tissues and how you maintain them, even more importantly, how you maintain tissues and how you make them resistant to failures, these issues of how cells sense their environment and respond to it are critical. One thing that we're going to talk about today in a very naive way, not with molecular species, is this idea that cell-cell contact generally inhibits proliferation, and that often goes under the name of contact inhibition. And we're definitely going to, by the end of the class, talk about growth factors and their role in promoting cell cycle. When we deal with cell cycle in reality, cells don't go from being undivided to divided instantly. The cell cycle is pretty complicated. It has a lot of steps. And during those steps, the cell first has to synthesize proteins. It has to grow. It then has to synthesize the DNA to copy its chromosomes. Those chromosomes have to align and separate from each other. After that separation has happened, there has to be a new cell membrane, and there's a physical division of the cell. What we call mitosis really is just the actual division of the cell, but even that takes quite a few steps. We have prophase, metaphase, anaphase, telophase, and that is inside of this longer cycle. We're going to approximate this in a very crude way by thinking that there is a growth phase that takes a lot of time, and that cell division happens instantaneously. And for a lot of purposes, we can get away with that. But there also are times when you can't get away with it at all, and we have to think again about what we're going to do. Because in especially early development, but also in some cell types in adults, you can have things happen like you can have the nuclear DNA replicate, you can have chromosome replication without the separation of the cell into multiple cells. When that happens, you have what are called syncytial cells, which have more than one nucleus. That may sound exotic, but if you go into your liver, the hepatocytes typically have two nuclei. So they've started cell division but not divided. Your myofibers, your muscle, is even more exotic. Your muscle fibers have lots and lots of nuclei. Those actually start out as separate cells that have fused together. Cell fusion in general is a pathological process, but it's not pathological in your muscles. Your muscles depend on it. And in your muscles, it's even possible to trigger the reversal of that, where the muscles fibers separate into individual cells that can then re-enter cell cycle. That's not normal, at least in humans. So all of this complexity is something to keep in the back of your mind, and be aware that we're ignoring it today. Another thing that comes up that's worth mentioning is that this whole elaborate series of steps that have to happen for a cell to replicate have lots of mistakes occur. And in general, as I mentioned in the context of infection, biology's strategy for dealing with mistakes is, in the first place, if the mistake is minor, plow through it. If the mistake is major, the one thing I don't want is for a cell to divide continuously without respecting its environment. You don't want it to run through stop signs. If it does, you have a tumor at best and a cancer at worst. And so all of these different phases, and there are many in cell cycle, have what are called checkpoints, which determine whether the cell can proceed from one phase to another. And the molecular basis of these checkpoints is complicated. They're all different. If you look at some of the models for cell cycle that are available in something like Biomodels database, some of these are modeled. Some of them are still not well understood. But the critical ones that we probably are going to have to deal with is entering into S. When you start S, you're more or less committed to going through the whole mitotic cycle. M, am I going to divide or not? And the exit for M, have I actually divided successfully or not? Typically, if the checkpoint fails, one of two things will happen. Either the cell arrests, so cell cycle stops, but the cell survives, or more commonly, it'll trigger apoptosis, the cell will die. It'll say something went wrong and I want to die. And one of the key reasons that mutations accumulate in cancers later on is that these checkpoints get blasted through, and so improper chromosome separation, improper DNA copying doesn't lead to cell death. Again, we're not going to address these things in detail, but these models exist and can be easily put into CompuCell models if you want. So for today, we're going to go back to something we did the other day in a little more detail, which was how to make cells grow in CompuCell. And then we're going to also think about how to make them divide. And that was in the homework. And so if everybody is all comfortable with it, I guess I could skip this, but I want to go over it at least quickly. And please feel free to ask for more detail if there's anything that's not clear about how this is done. Again, the rates of both cell division and cell growth and cell division depend on all sorts of things. We've mentioned how much nutrient the cell can absorb. If it doesn't have material to build with and energy to build with, it can't grow. How fast the cell can convert that nutrient into new mass, which is slightly different from the rate at which it can take it up. The efficiency of that conversion, the presence of specific growth factors or growth inhibitors, contact with other cells on the substrate, the internal state of the cell, and then, for example, in a negative sense, usually, although some toxins actually promote cell growth and division, whether there are things in the environment that are perturbing it. In our world, cells will grow according to a simple rule and divide according to a simple rule. So we're going to neglect all of that complexity. And of course, our cells don't have all this beautiful macromolecular machinery that is responsible for chromosome copying, organization, and separation. So the easiest way to have a cell grow in CompuCell is using the target volume. In fact, it's the only way you can have a cell grow, because the only way we can control the volume of the cell is through the target volume. And the simplest exercise that we could do, which we did, and we'll do again, is to have the target volume of each cell increase at a constant rate in time. We can't change the volume of the cell directly because we don't know what is happening to the cell from the outside. So we don't have the ability to directly change the volume. We can only change the target volume. To do that, we have to change the volume plug-in to this form that we discussed, just volume, to say that the cell does have a volume. And then we specify the initial volume, the target volume of the cell, and its compressibility, inverse compressibility. And then here in our step function, we make that cell's target volume go up. And here we're having it go up at a constant rate per time. So homework 4.1 started out by asking you to implement a cell growth simulation of this kind. And I'm going to ask people to do that together today, and we'll work up from that. So I'd like everybody to fire up CompuCell, whether it's on NanoHub or on your desktop. And the first exercise, which is 5.2, which was homework 4.1, was create a simulation on 200 by 200 lattice, square lattice. Could be 256, the size isn't that important. you want it to be big enough that you're going to get quite a few cells by the time we're done, so you don't want it to be too small. Use periodic boundary conditions in the X and Y directions. No flux in the Z direction. Use third neighbor interactions in the blob initializer. And to begin with, create one cell type. You call it cell, if you like. And give the cells contact, volume local flex, surface local flex, and growth as properties. And then figure out how to use blob initializer to create a single cell center of simulation. And I think because I pulled these slides from different places and I was trying to go back and make them compatible with the homework, there may be some inconsistencies when I say make the cell this big, or this big. I suggested making the initial target volume of cell 25, lambda volume of 3. And then there was a good point made in a comment on the homework. I said have the cell grow at a constant rate so it doubles in volume every so often. And that, and somebody pointed out that, of course, the cell may double once, growing at a constant rate, but I would have to double the growth rate to make it double at the same time a second time. And the reason it was written that way was because initially it was written in the cell division. So the cell grew and divided and then grew and divided. So the doubling volume was a fixed amount. Whereas in a non-dividing cell, it didn't make sense. So I appreciate somebody catching that. And then it's useful to create a plot window that'll display the pressure in the cell versus time. And so let's do that together. So let me do it myself. I'll create a cell growth simulation. I said 200, so I guess I need to change the size of the lattice. Periodic boundary conditions. I said third neighbor interactions. Blob initializer. One type of cell. No chemical fields. Contact. Growth. Volume local flex. Volume surface local flex. Create the simulation. I said we want a single cell. So I have to go to the XML and change blob initializer so that the radius of my blob is small compared to the cell size. I make my radius something like 5, maybe 4. Otherwise, I don't need to change anything there. And then in the cell growth steppable, I said set the initial target volume to 25 and lambda volume to 3. And then it asked us to have the cells grow at a rate of 25 voxels per 500 Monte Carlo steps. So that would be, let's see, I want it to be, I don't know quite why I picked those numbers. 50 for 1,000, so 0.05 per Monte Carlo step. And I asked to create a plot window to display the pressure. So let me create a plot window. To create a pressure. pressure. And at the moment, there's only one cell, so I don't have to worry about averaging or anything like that. And I put this in the wrong place. I need to put it in the start function, not the step function. And then I will, in the step function, I will plot pressure. And that should be in the, that should be in the loop. Okay. And I can walk through this with people, assuming that I didn't make a mistake, which I guess I did.
So I don't know if this is what's causing it, but when I was working on the homework, because it was establishing it in different steppables, I had to merge them into one.
Ah, yes. Well, I just did exactly that. So, yes. So the plot window has to be created in the place you're going to use it. So let me move it.
And that actually threw me off a little bit in the documentation, just because it says to make sure you put that initialization
Yeah.
in the start section. So are you just about to put it in the growth steppable class?
I was.
Okay. Okay.
Or I could put the display into the other.
Okay.
So I have a choice. I can put the see, I don't have a step function defined here. So I'd have to either create a step function in the outside of the growth steppable into steppable base, or I have to put it here. So, and actually I don't know, Giuliano, if I put the plot initialization into the init, what happens? Well, let's try it, see what happens.
And then, is there anything wrong with removing, like commenting out the first 3 lines of the second class just to make it a singular steppable?
You certainly don't have to put, mitosis has to be at its own steppable. Or else you have to put everything into the mitosis steppable because it is fundamentally a different structure. Growth doesn't have anything special about it.
Okay.
I don't know what will happen if I do that. I haven't tried it. Will it complain about the init? Looks like it didn't like it. Anybody else have an issue there? Giuliano, you know about that?
Yep. No, I stepped out for a second. I didn't see what error occurred.
So the question was the way this is set up, growth steppable doesn't have a start function.
Right.
And so there's no place obviously to put the plot.
Right. Yeah, okay. Yeah.
And so I asked the question because I had never tried it. What happens if you put the plot initialization in the init? And the answer is it crashes.
Okay. Yeah, that makes sense. But you can create a start function.
Now that would be the easiest, probably the easiest thing to do.
Yep.
Which is,
It's what I'm doing over here.
now do you need anything in it besides literally the start line?
Nope. You just just the start line.
I just put in the wrong. I put in the wrong order. It has to go here.
Yeah.
Yeah. Yeah.
Yep, that should work.
So that might actually be something in that growth steppable. You should probably add an empty start function.
Mm-hmm.
The default. Did anybody else have an issue with this? And if so, how did they solve it? I'm not sure why I have pressure twice. I probably instantiated it. Oh, because I didn't take it out of the. Okay, does everybody have that? Did anybody need help with that? And the rate at which I have the cell growing is probably too slow. I should probably speed it up. But I can see the cell grow a little bit. And the question is, what happens to the pressure of the cell versus time? And the answer is not much.
For me, it starts off not doing much and then starts to reach higher and higher negative values. So it's increasing. I'm not sure if I did something wrong.
My guess is that you have the surface energy turned on.
Yes.
So that's the next step. So you beat it.
Okay. Okay.
Anybody else have any questions about this? Let me come back to the code. So again, creating the simulation here, I had it as 100 by 100, blob initializer, pixel copy range of 3, create a cell, contact, volume local flex, surface local flex, growth, center of mass. Edited the initializer to create a radius 5 domain, so there's only one cell to begin with. Then added the initial volumes of the cell here. And then had the cell grow. And I have it growing here probably too slowly. Let's try 25 divided by 500. And then adding the plot wind and the plot of the pressure. So the thing that was pointed out by Connor is that you need to create a start function to put the plot window creation in. And to do that, you type def start parenthesis self close parenthesis colon. That's all you need. And I agree that either growth steppable should have a start function by default inside of it, or we need to document better that you need to do that. Thank you for catching that. Okay. So now that we've got that working, why don't we add also the ability to display the volume of the cell and its surface tension as well. And it's going to be up to you to decide whether you want to put those in the same plot window or not. It's going to depend a little bit on the scale. And I don't know a priori what the numbers are going to look like. So you might want to create separate plot windows for those. Okay, so everybody take a minute or two to do that. So the question was, I asked for a tracking field to monitor the pressure and the surface tension. Maybe we'll turn the tracking field on later. I guess we can do that too. So I'll add the tracking field as well. Remember to do the tracking field. The tracking field, unlike the plots, has to go in the init. Extra fields, automatic tracking, scalar attribute. We're going to have two of them, one for pressure and one for surface tension. And remember, with tracking fields, you have to copy the value into the cell. So in the step function, we're going to need to add in our loop cell dot dict pressure equals cell dot pressure cell dot dict tension equals cell dot tension, surface tension. Okay, so does anybody want to tell me what you see when you run the simulation? In the interest of time, let's skip this plotting the target volume versus the actual volume. That's something you can add yourself, and it basically is another way of monitoring the pressure. But essentially what I see here is the pressure stays roughly constant. There is some pressure because there's a positive energy between the cell and the medium, so there's some compression. And pressure is measured backwards, so inward pressure shows up as a negative number here. And the volume is increasing essentially linearly in time, which is what we've asked it to do. Okay, any questions about that so far? Does that everybody get it? Does somebody need help with that? So we see the cell grow. In fact, the growth rate I picked was very slow. Let me pick a faster growth rate so that I don't have to wait forever for things to happen. And now I can see my cell growing as I run the simulation. Not the most exciting simulation in the world. So the next thing I'd like you to do is turn on the surface area constraint. We loaded the plugin, but we didn't enable it. So add the lines cell.target surface is 20, cell.lambda volume is 3. And what's going to happen now? What will you see? I think, Connor, this may be already where you were to begin with. So does anybody want to predict what's going to happen when we do this?
Hey, I just want to tell you, there is, I think, just a mistyping. Instead of lambda volume there, I think it should be lambda surface, shouldn't it?
Oh, yes, thank you. I'll fix that. That is a classic cut and paste error. Okay. So what happens when you run that? How is it going to be different?
For me, it's that the pressure is continuously going more into the negative while surface tension and volume keep getting higher. And my cell also takes on a rigid shape that originally made me think I did it wrong, but I'm not totally sure. No, okay, yeah, that's what I have.
And the reason here is that you're limited by the surface area. And so you're basically building a golf ball or something, or a basketball that's very rigid, that's under high hydrostatic pressure. With the caveat that because we're on a lattice, instead of getting a circle, we get a square, because that's got the smallest surface area per unit volume. And you'll notice it still grows a bit because we're pushing against that surface. But yes, now you see the pressure going down. And the sign of the pressure, again, is a bit backwards. We probably should put a minus sign before we plot the pressure, because our instinct is that the pressure should be outward directed, and it's inward directed in the definition. You'll see the surface tension keeps going up now. because we're stretching the surface more. Okay, does anybody need more time for this one, or does everybody have that working? So we get that, which we saw. And so if we're going to have the surface area constrained along with the volume, then we have to have a sense of what's reasonable. And so we're going to have to adjust the target surface in a variety, one way or another, to keep things constant. One way to do it would be to adjust the target surface to keep the surface tension constant. Another would be to adjust the target surface to keep the pressure constant. Both of those are possible. And again, because of time, I don't think we're going to explore all of these options, but they're in the homework. Let's try doing this first one, which is adjust the target surface to keep the surface tension constant. How is the surface tension defined? The surface tension is given by the lambda surface times the difference between the actual and the target surface. And so if I adjust it so that the target surface is updated to be the current surface minus something, then I'm keeping the surface tension constant. Excuse me. Keeping the actually, I'm not sure. Was I muted when I just said, when I explained that? I thought I'm okay. I thought I
No,
muted
no.
the cough, and then it wound up. Then I didn't. Okay. So keeping the pressure constant actually makes more sense, but it's a little bit more work. So let's try adjusting the surface tension, or the surface, so that we keep the surface tension constant. So to do that, anybody have a suggestion for what we need to do?
As you said, we should decrease the target surface by an amount. I'm trying to figure out the amount. The same rate of growth, maybe?
Well, so we want to increase the target surface
Oh, okay.
because the cell's growing. So to keep the tension constant, I could come here, and I probably want to do it before rather than after here. And I could say cell dot target surface equals cell dot surface minus some number, n. And then the surface tension will always be 10 times 2, 20, times lambda surface, which is 3, so 60. So if I do that, then I should find that when I run this, the surface tension is always 60. We'll see if that's the case. Try that and see what you get. $60. And my cell is still pretty square, which means it's too much tension. So let's try adjusting it to see what's reasonable. Why don't you do that? Does everybody have something like this running? Why don't you try running that and then tell me what value of surface tension I should set so that the cell stays round. I know if I set it to 0, the cell will be round, but that's effectively turning it off. So what value could I use that would keep it round?
I did the same that we used for the growth, the same rate for the growth, and mine is circular.
So you use this
Yes, yeah.
for the target. So that's
Yep.
also a way you could do it. Yes. You could increment the target surface by the growth, and that would work. I'll try something like 2 here. See if that works. So there are a whole variety of ways you could make this work. The key thing to be aware of is that you need to keep adjusting the surface to match the volume. And you'll see pathologies where the cell will become rigid or square if you don't do that. Here probably I'm still too strong. Did anybody else come up with a different solution? Another way to do it would be to adjust the surface based on the actual volume. Here, I've said that the target surface is 2 times pi times the square root of the target volume. And that's basically trying to keep the pressure constant. A third way to do it is what Joel suggested, which would be, say, cell dot target surface plus equals 25 over 100, and then play with that increment to see what works. So they're not all exactly the same. If I increment target surface at a constant rate, that's going to increment faster than this would because this is going as the square root of the volume rather than the actual volume. So this is actually growing a little bit slower. So there are a variety of solutions that one can play with. Now, I've asked here, based on the homework, some additional things that I don't think will take time to do right now, because I want to keep going to cell division. Because that was the purpose of the class, was to get to cell division. That you can explore how to do this kind of setting. Okay. So now let's talk about cell division a little bit. So not every cell that grows divides, and cells can divide without growing. They have to synthesize DNA to divide, but they don't actually have to grow in volume. So early on in embryogenesis, after fertilization, the egg will divide a significant number of times without any significant amount of protein synthesis. Depending on the species, it may divide 10 times, 12 times. And so the cell volume may actually go down by a factor of 1,000 or more from the egg later on. So that is a situation where you have cell division without growth. Similarly, you can have growth without division in a variety of situations. A classic one would be some kinds of marine algae, where the whole algae is a single huge cell. It keeps growing and growing and growing. In that case, the nucleus is replicated, but the cell doesn't divide. And you also have cells like some of the cells in the cornea, which grow in time, but don't divide. So it's possible to have both. Again, as we pointed out, mitosis is an extended process, and it's not always complete. Cells can separate their chromosomes without separating their cytoplasm. And cells that partially divide and leave a bridge of cytoplasm between the two offspring are pretty common. Computationally, that's not something that we can really handle, because you either have two separate objects or you don't. So if we want to build something that's syncytial, which represents incomplete cell division, we'll typically do a cell division in CompuCell to two separate cells, and then we'll define some other additional properties that basically connect the cells back together. In reality, as we mentioned with checkpoints, the chemical signals that determine when a cell divides are very complicated. In our case, to begin with, we're going to use the simplest possible assumption that we can have, which is that we're going to have the cells divide when they reach a target volume, conceptually when they've doubled in volume from their initial state. When a cell divides, we're going to go from having one cell to having two cells. That means our simulation is now going to have an extra cell in it. We've added a cell to the simulation. And when we do that, we have to make a bunch of decisions, both biologically and computationally, about what's going on. In the first place, we have to say what parts of the parent cell get transferred to the daughter cell. Is the division symmetrical? So half of the cell volume goes to one cell and half to the other, or asymmetrical? Both of those things occur biologically. Is the division in a particular direction relative either to the external world or to the elongation of the cell? Very often cells divide along their long axis, but not always. The default in CompuCell is going to be that cells divide along a random plane, but we have to make a decision about that. Our cells have a lot of attributes. They have target volumes, target surfaces, and so on. We have to decide how those attributes are assigned to the daughters. The key one is going to be target volume and target surface. Conceptually, we could think of target volume as representing the mass of the cell, and the target surface could represent the membrane of the cell. And we'd expect, if we have a symmetric division, that the daughter cell would inherit half of the volume and half of the membrane. Notice that that won't quite work, because the scaling, when we change the volume to keep the pressure the same, doesn't divide the membrane in two. And so if we do that repeatedly, we'll discover that the membrane area gets to be off. Target surface will get to be off from what we expect. And so that's a reason to use a pressure-based or tension-based assignment of target surface. We need to know whether the daughter cells are of the same type or not. Very often a cell will divide and spin off cells of a different type. When stem cells divide, typically one offspring is another stem cell, the other one is a differentiated cell of some kind. And we also have to make a decision about whether the parent cell or both cells continue to proliferate or not. Biologically, it's pretty common as a safety measure for cells that after a cell division, by default, the cell stops. It exits cell cycle. So the cell will divide, and then wait for some active signal to start dividing again. But that's not always true. Again, in the example of early embryogenesis, you have a whole series of cell divisions and cell cleavages which are continuous. There isn't a stop signal in that case. And so those are all decisions that are fundamentally biological decisions, but that translate very directly into computational decisions. We've been dealing with the annoyance of the growth steppable. Now we have to deal with another minor annoyance, which is that to do cell division in CompuCell, we have to use a special kind of steppable, and that steppable is called a mitosis steppable. And the reason is that that particular steppable has loaded functions that are going to be associated with dividing the cells, which is fairly complicated to do, and also for the inheritance of the properties from the parent to the daughter. And again, we're going to find that unless we explicitly have the daughter inherit, it doesn't. So the daughter will have no properties, and we'll do that as an experiment to see what happens. It's not very pretty. Typically, we're going to have some event that's going to trigger cell division. And one thing that we're going to have to do is that we don't want to be doing cell division and deciding what's going to divide at the same time, which can cause problems, because then you have what if you have a cell that really needs to divide twice? You don't want it starting to divide in the middle of things. And so what we're going to typically do is we're going to sweep all of the cells in our simulation, or the ones that are in cell cycle. We're going to ask the question, should they divide or not? We're going to make a list of the ones that are going to divide and then pass it to this function that's going to do the division. And so we're not going to do division on the fly. We're going to do it all at once in clumps. Okay. If I look at the code, the mitosis class, if I add it, and we'll do that in a second, you'll see class mitosis steppable, mitosis steppable base. We don't usually pay attention to those, but if you'll notice, it's a different name if you look carefully. Again, there's no start function by default here. You can add it. Def step, we're going to create an empty list of cells to divide. And here we're going to have a very simple rule. If the cell volume is bigger than 50, we add the cell to our list of cells to divide using an append, because it's a list. Then once we've gone through all the cells, we go through the list of cells to divide, and we make them divide. Here we have self.divide cell, random orientation of cell. And we'll see that we could have the cells divide in a specific direction along their major axis or their minor axis. The next thing that we have to do is say what the daughter cell and the parent cell do after division. By default, the parent cell doesn't change its attributes at all, and the daughter cell doesn't have any attributes, neither one of which is what we want. And so there is a function which is called after division, called update attributes. And typically the first thing we're going to do is say self.parent cell.target volume divide equals 2. We're going to say it's a symmetrical division. And then there's a very powerful function which took a long time to get written, like years, called self.clone parent to child. And that's going to copy all of the properties of the parent onto the child. And it should actually copy everything, including the dictionaries and everything else. I won't promise that it absolutely always copies everything, but it's supposed to. It uses a function called deep copy in Python, which is slow but very powerful. It's possible also to only clone certain attributes of the parent. We typically don't do that. And then the default in the boilerplate that will be pasted in does something that's not going to work in the context of this simulation because we only have one cell type, which it says if the parent was of type 1, the child is of type 2, the parent was of type 2, it's going to make it of type 1. And this will cause our simulation to crash if we've only defined a single cell type to begin with. So we're going to have to edit that out to fix that. So let's do that. Let's go back to our simulation. And we've been typically creating simulations using Wizard, and we could do that and go back and redo things with Wizard. Here we are going to do something we haven't done so far, which is we're going to add a steppable to our simulation. And this is also something that didn't exist in CompuCell until fairly recently. There are two syntactic ways of doing that. I can click on in my navigation bar, I can click on my steppable name. I can right click and select Add Steppable. Or I can go to the Python drop-down menu that I'm used to using and go to the very bottom and click Add Steppable. And that will pop up a window. And this will give me a set of choices about what I want with that steppable. And I need to give it a name. I'll just call it mitosis here. I need to say how frequently it's going to run by default. The default's once per Monte Carlo step, but I could run it less often if it's going to do something like display something or save output. I have to say what kind of steppable it is. Generic, mitosis, which is the one that I'm going to use. If you have subcells, if you have cells that are composed of compartments, cell division gets really complicated. So if I have a cell that has a nuclear compartment, a cytoplasmic compartment, a lamellipodium compartment, then dividing the cell, I have to divide the cell as a whole, but I have to divide the compartments as well. And the way that that's done is pretty elaborate. It works very nicely, but it's elaborate. It's also quite slow. And so cluster mitosis is something you use for that. And then in principle, I can choose to add visualization field, tracking fields here if I want to, although I'm not going to do that. So go through, select steppable name mitosis. Make sure you click mitosis for steppable type and hit OK. And if you now look at your code, you will see at the end of the code, class, mitosis, steppable, and so on, precisely what I walked you through a minute ago. So everybody, please do that. If anybody has trouble, raise your hand and we'll walk you through it. I'll leave this on the screen for a minute until everybody's caught up. Okay. Reminder, which I mentioned earlier, when you have multiple steppables and you need to pass information between them, you have this self.steppable underscore shared underscore VARS, which is a dictionary that's a global dictionary. And so if you want to pass information between steppables, you can put it in that dictionary, and it's accessible to everything.
When we use that, do you have to include self in the variable name, or is it just the variable name? Like if you wanted to plot a window that you initialized in a different steppable and wanted to plot a variable in another one, would the variable be self.plotWin or just plot win?
So self, it's a little mysterious why this is self, because self normally shares within the class.
Mm-hmm.
And so normally it's within the steppable is self. And so adding a self dot doesn't get you out of the class. I mean, it doesn't get you out of the class.
No, I'm talking about in the key of the shared variable dictionary, like in all caps variable. Do you have to include this self?
No.
Okay.
No, this is a string.
Okay.
So this is a name. I mean, it could be a numeric. It's literally an alphanumeric string.
Okay.
And that's exactly the value of this. And when I've got it capitalized here, it doesn't have to be capitalized. I just wanted to show that it actually is something that you change rather than something that's left in that form. There are globals in Python. The one thing that you run into with globals in Python is that you have to remind Python that they're globals. And so in many languages, once you've declared a variable a global somewhere at the beginning of your code, it's available everywhere. In Python, you have to keep reminding Python that it's a global, because you can, if you declare a global at the beginning of your Python code, you can access that variable for read anywhere, but you can't write to it. And so you have to keep re-globaling it every time you want to write to it, which is rather annoying. For parameters, it's not a problem. For variables, it's a problem. Well, it's an easy way to make type. It's an easy way to create a bug. It's not the end of the world. It works, but it's easy to forget. It can create. Python is a language that's very powerful. It's also a language that's pretty good at writing bugs that are hard to track. And that particular issue with globals, that if you don't re-globalize them, they can be read but not written to, is a classic way to make hard to find mistakes. This works. Okay. So again, walking through the code, we have a step function. Well, we have our mitosis steppable base here, which we won't play with. In our step function, we create an empty list, which we're going to put the list of cells to divide. We're going to check if the volume of the cell is bigger than 50. We add it to our list with an append. And if you're not a Python person, lists and append are one of the relatively limited Pythonic things you have to learn. And we'll try to go over them multiple times. Once we've made our list, we go through the list. And the reason we do not want to do the divide in this loop is that a big no-no in Python is to change the thing you're iterating, the list you're iterating over while you're iterating. And if we divide a cell, we change self.cellList. And that means our iteration can have problems. So that's why we separate them. And then we go through cells to divide. Here we're going to divide it with random orientation. We then have our definition update attributes, which gets called every time we divide. We assign half of our volume to the parent, we clone the parent. And the daughter, by doing this, by changing the parent volume first, the daughter inherits half the volume too. I could have the daughter clone first and then change both volumes afterwards if I wanted to. And then the one thing that I have to do is I don't want to change the type of my cell because I don't have another type. So I can either delete this or comment it out. I'll just comment it out. I don't want to change the type. Okay. So why doesn't everybody try that? Let's see what happens. Did I crash CompuSol? Looks like it. Did it work for somebody or did it crash? Joel says it works. I don't think it's my typo. I think it's NanoHub.
Yeah, I can get mine to run locally as well. So.
Anybody else? Good. So, did people get something that looks a bit like this? For your simulation. Let's come back just for a second. If you want to operate, there's a reasonable question, which is which cell is the parent cell and which cell is the daughter cell or the child cell? Computationally, one of the cells will have the ID number of the parent, and so its identity didn't change. And there will be a new cell created with a new ID number, which is the daughter. Self.parent cell, only in that context of the mitosis steppable, identifies the parent, and self.child cell identifies the daughter. And so if you need to do something specifically to one of the two cells, you have access to do that. And you'll notice, for example, if you colored the cells, that one of the parent, well, the parent would keep its color and the daughter would change. So in this case, we've already done this. I'm going to skip ahead. In this simulation, I was going to add a cell type. We can do that later. I don't want to take the time. This simulation is the same simulation now with two cell types where that switching between cell types was kept. So that's the difference. Otherwise, the simulations are identical. Also here, I've averaged the target volumes and the pressures instead of looking at all of them. But they're not very different. What I see here is that the volume of the cells all goes down simultaneously. They grow, they go down, they grow, they go down, they grow, they go down, they grow. Why do you think that they're not in phase here at the end?
Is it might be because of the contact in between? Do you have a lot of cells in contact?
What was the rule for division? All the cells are growing at a constant rate, so the target volume of the cells is increasing at the same rate. But what was our rule for cell division here?
If it gets to a certain volume.
Sorry, I don't like scrolling when you have to watch, but that was our rule for volume for cell division. Now, is the volume something we control, or is it something that comes out of the simulation?
We are providing the rate, right? For it to.
We're providing the rate to the target volume. But as we've seen, the actual volume will depend on the situation the cell finds itself in.
So is it the ones that are in the center are being compressed so that they aren't having the same rate of division?
That's right. So let's let this continue to run. It's taking its time here. Maybe I should have had them grow a little faster.
I think your steps stopped. Oh, there you go. You got 10. You see, it's very slow.
So can anybody tell me what happens when the cells run into the wall?
We have periodic boundaries on, right? In this case?
Right. So they run into each other, run out of room. What happens when we run out of room?
Wouldn't we see the case similar to what's happening in the center be more widespread across all of it?
Will any cells divide?
I think they should reach a point where they stop, shouldn't they? Yeah.
The volume can't go up. If my target volume is 50 and my volume of my lattice is 200 by 200. So 200 by 200 is, let's see, 40,000. 40,000 divided by 50 is 4,000 divided by 5. So when I get to 800 cells, if I have more than 800 cells, none of them can be over 50. And so they'll never divide. Now, in fact, it's possible in some simulations to have clusters of cells that will collapse because of the pressure and disappear. In that case, you can release space for cells to divide. And that happens in some tumor simulations, some cancer simulations. It actually happens in real cancers. When you remove tumors, not always, but many solid tumors are nutrient limited or space limited. And that means that their rate of cell division is actually fairly low. And if you then go in and either kill them partly with chemotherapy or radiation, or remove them with surgery, and you don't get all of it, those cells that are left behind suddenly have plenty of space and plenty of nutrient and divide like mad. And then you get very, very rapid somatic evolution. And that's how you develop resistance. You develop resistance to therapy anyway, probably, but the rate would be much lower because the rate of cell division would be lower. And so that leads to this paradox that treating cancers can actually make them worse. So I apologize for keeping people waiting here. But what we should see, and I'm not plotting the target volume here in my graph. I didn't create that one. But if I were to run this as the instructions were given, and we're almost out of time, so I don't want to do that now, we would see that the average volume of the cells would keep going up, but there are no cell divisions because there's no room for the cells to grow, and therefore the cells never actually reach a target volume, an actual volume of 50. And so the system gets stuck. The pressure keeps going up because I keep adding material to the cells and they can't grow, so they get more and more compressed. The system will get more and more rigid. You'll see fewer and fewer fluctuations, but the pattern will essentially lock in place. I'm not sure why NanoHub has gotten so slow right at the moment. This isn't that demanding a simulation that it should be crashing. There are a couple of other exercises that I wanted to do. They're mostly in the homework. So I think we'll keep that homework from last week if people haven't. How many people did all of the homework for this week? No? One.
I made it partway through. Like for each of the 3 problems, I made it partway through and then ended up getting stuck. So I have everything like partially completed.
Okay.
Same here. I have like at least the first one done on all 3.
Okay. So it sounds like for people, so I think what we'll do is, given where we got to in class, we'll carry over the homework assignment. You'll have the 2 extra questions, which is assign yourself something to do for your project and report on what you did. And then for people who did all of it, congratulations. And I would suggest that you could go back, if you want to explore this more, there's a lot to explore with cell division. There are lots of interesting paradoxes with cell division. How does oriented cell division change what you get? Can do it less than you would expect, actually, because these issues are pretty fluid. But that's an interesting question. Another one, and this is a big difference, is if you have the cells divide not when they reach a volume of 50, but when they reach a target volume of 50, you're going to get a totally different result, because in this case, the cells will continue to divide whether or not there's space. In this case, every 100 Monte Carlo steps or whatever you define, you have twice as many cells. And so what you'll see is that the cells initially will grow the same. It'll look identical. As it runs out of space, however, in this case, the cells will look like they freeze. They won't grow anymore, and the pressure will keep going up. In the other case, and it'll go up linearly. In the other case, the cells will get smaller and smaller because they'll divide by 2 in their volume every so often. Eventually, some of them will disappear, so it won't be quite right. And the pressure will go up not linearly, but exponentially. Every time there's a doubling, the pressure will double. And so you'll see a stepwise function where the pressure goes up linearly, then doubles, and then this gets steeper and steeper as you go. And so this comparison of, seems like, why should it be different whether I divide by volume or target volume? Well, they're similar. But in fact, in these slightly pathological circumstances, you get a very different result. And that kind of thing does happen. Famously, not in division, and it's not really what a biologist would call growth. A biologist's definition of growth is not the volume increases. A biologist's definition of growth is that there's protein synthesis that makes the cell get bigger in a particular way, not that water is coming into the cell. From a simulation point of view, what we care about is the volume. From a biological point of view, it matters what the volume is composed of. But as a classic example of that is the famous, maybe just so story, maybe urban legend, that if you drill a hole in a rock and you put beans in it, dried beans, and you pour water on them, you crack the rock. So that's an example where essentially the hydrostatic pressure of those beans is large enough to crack rock. So in one case, the volume, the target volume and the actual volume are very close together. And so there are some interesting differences that you can explore there. And I hope that people will take that's right, that's exactly right. And so I hope people who have finished the formal homework problems may explore some. I'll have the slide deck is available online. So if you want to walk through some of these questions and see how things differ, I hope you'll find it interesting. This is showing that exponential growth of pressure at the end when you have the fixed doubling. Where you divide by target volume rather than actual volume. Okay, well, we'll call it a night there. Are there any questions before we break?
Yeah, do you want us to upload like what we showed you today for our like project proposals?
Yes, please do that.
Okay, but I see there's two. There's preliminary proposal write-up and presentations, and then there's mini project preliminary write-up. I don't really understand the difference between those two. Or
they're both due today?
Yeah.
That probably is a mistake. I think this happened before. One of those may be a ghost from last year.
Okay.
Because I noticed that for homework 3, somebody uploaded the homework into last year's. I didn't realize it. It looks like Canvas tried to be smart and updated all of the old assignments to new due dates.
Mm-hmm.
And so then when I created a new assignment, I didn't realize that there was a ghost old assignment.
Okay.
Should
I just pick one then, or?
Pick one, yes.
Okay.
And I'm sorry about that. I'll try to prune that. And the other thing I was going to say, just as a comment on the homeworks from last week, which is that when I ask you to show something, always in your write-ups, give the parameter values you used. If I say what happens when you change the surface energy, contact energy, or what happens when you change lambda volume, always in your write-up when you show a picture, have under the picture the value of these parameters was this. Because there's a lot of ambiguity, and it's good to. Now, of course, here I've done something where I'm showing a picture without having labeled it. In this case, I plead guilty to not following my own advice. But in the homeworks, always when you give a report of a simulation, give the parameter values that you used in the simulation. And ideally, also, please do zip. I know Canvas isn't very friendly it doesn't let you upload multiple files typically for one assignment. So if you want to upload your code, you'll have to do 2 zips. Or actually you don't have to do 2 zips. But you have to zip everything together and upload the zip so that your simulation code is included. if everything works, that's fine. But if there's a problem with your simulation code, you say, what happened? And I don't have the code, I can't debug it. I can't promise I'll run everybody's code and try to debug it for you. But I will at least try to run it, usually. And if you don't include the code, I can't run it. The disadvantage of that is then the speed grader in Canvas doesn't work. Because if you upload a zip file, I have to download it and unzip it to look at it. But I think it's still better to do that. The other way I could do it, I guess, would be to create a second parallel assignment where you uploaded the code to the second assignment, but that's getting pretty cumbersome. But do please remember to include the parameters when you give your results. If you show me a picture and you say this was for a growth rate of 10, this was for a growth rate of 50, I want to know what those rates were. Especially because things like neighbor ranges that we went over, lattice types, there are a lot of parameters that might be different and that do affect the simulation. And so it's important to know what those are. Anything else? That was a good point. Everybody okay? Well, thank you again. I appreciate you. Every week you come back. I am grateful to you. I do appreciate it. And I guess we will be, some of you I'll be seeing for meetings with Gary and Roland and TJ. And so please keep going and contact me if there are any questions about homework, methodologies, or projects. And with that, I guess we're done for tonight. Thank you.